Hello, this is Dr. Morris continuing with the lecture series for Anatomy and Physiology 1. Today the lecture will discuss basically a review of the histological slides for the histological or tissue level organization for ANP1. Again, this is a review of histological slides. Previously in the lecture, we talked about tissue level organization, and I went over some of the specific tissue types that you would be responsible for identifying. I have prepared a series of selected slides here off the website that's also a part of your lab assignment, which has been posted on the blackboard. One of the types of tissues that you will be expected to identify for the gross anatomy examination would be for you to identify a simple cuboidal epithelial cell. Now, an example of a question I would ask, I would show you this slide and I would ask you to identify the type of epithelial cell that is located in the collecting duct of the kidney. Your response should be simple cuboidal epithelial cell. That's why it's important for you not only to be able to recognize these tissues, but to also know what the function of the epithelial tissue is, and to know its location. So simply by me telling you in the collecting duct of the kidney, we know that simple cuboidal epithelial tissue is located in the collecting ducts of the kidneys, whether that's in the renal tubules or the distal loop of Henle. All of those structures that are located in the kidneys typically are lined with simple cuboidal epithelial cells. Again, as you remember my description of cuboidal cells previously, these are basically square-like cells. As you can see, they have very prominent nuclei that are usually centered into the cell. But these are better pictures of cuboidal cells over here, located in this portion of the slide. Again, if you go to the website in your lab assignment, it takes you to the University of Kansas Histological Lab. And there's a series of prepared histological slides. Some that have been selected for you are for simple cuboidal epithelial cells. This is another example of cuboidal tissue. Now this one is different. This is the stratified cuboidal epithelial tissue. Again, we see this in some of the glandular operations. Remember, stratified means that the cells are stacked upon each other. Again, prominent nuclei. These are square-like cells, but they are stacked upon each other. A little more difficult to recognize than some of the other cuboidal representations, but this is stratified cuboidal tissue. This slide represents simple columnar epithelial tissue. If I asked you to identify cells that are located in the digestive tract, the cells that we have talked about that are located in the digestive tract are columnar cells. Now there is some columnar cells that are located in the lungs as well, but for the purpose of this examination, the typical reference to columnar epithelial tissue location I would be looking for would be in the digestive tract. Also, to make it a little bit easier, I would also ask you to identify surface appendages. So if you see columnar cells with, say, microvilli as surface appendages, then you would know that this was in the digestive tract. Microvilli, remember, are finger-like projections that increase the surface area. You see that primarily in regions of the body that are involved in absorption, such as in the GI tract. Again, this is simple columnar epithelial tissue. Again, this is in a glandular structure, but again, the pattern is very identifiable. These long column-like cells, large prominent nuclei. The nuclei tend to gravitate more toward the basal lamina, see them more at the base of the cell. But again, this is simple columnar tissue. Again, this is simple columnar epithelial tissue. This is the ideal picture. This is the one that I would probably, or one similar to this, I would show you on an examination. I would ask you, I would identify first that this tissue is located in the digestive tract, involved in absorption. Please identify this cell type. You would tell me simple columnar epithelium. Then I would also ask you to identify the surface appendage. Again, if you know that this is in the digestive system, in the GI tract, say small intestines, the surface appendage would have to be the microvilli as previously described. Now, this is pseudostratified columnar epithelial tissue, ciliated. Let me repeat that. Pseudostratified columnar epithelial tissue, ciliated. Now, pseudostratified columnar epithelial tissue is located in two locations. One in the respiratory tract, say the trachea, or two in the male or female reproductive system. Remember that the cilia are surface appendages primarily responsible for moving substances across its surface. In the respiratory center, there's a layer of mucus that's over this ciliated structure. The mucus traps particulate matter, and in a wave-like motion, the cilia actually helps to throw the mucus outside the respiratory tract. So a person would either cough it up as phlegm or swallow it where it would enter the digestive tract, and whatever the debris or bacteria or viruses would be killed by the digestive enzymes in the stomach. So this is the cilia. Now this is pseudostratified columnar epithelium because this gives you the appearance that the columnar cells are stacked upon each other. Actually, they are not. But it gives you that appearance, and thus the name pseudostratified columnar epithelium. But again, the key items here, I would tell you where it's located at. I would tell you this particular cell is located in the male reproductive system. It has surface appendages. Please identify this cell type. You would be able to tell me pseudostratified columnar epithelium. I would then ask you to identify this surface appendage, and you would tell me cilia. I could similarly ask you, this is pseudostratified columnar epithelial tissue, ciliated. Give me two systems in the human body where these cells would be located. And you would tell me the male or female reproductive system and the respiratory system. Okay? This is in the bronchial tube. Now, obviously, this is not as high of a magnification as the previous slide. But again, if I told you this section of tissue is taken from the bronchial tubes, this is a ciliated border, please identify what epithelial tissue would need to be here lining this respiratory passageway. Again, you would say pseudostratified columnar ciliated epithelial tissue. Now, transitional epithelial tissue exists in the urinary bladder. Again, it exists there because it's highly expandable. It's stratified epithelium. This is referred to as transitional epithelium in the bladder. And here's a classic picture of that transitional epithelium in the urinary bladder. Again, I'm giving you the location of this tissue, which I would do on examination. I would show you this exact slide, and I would ask, this particular tissue is located in the urinary bladder. Its function is to allow expandability of the organ system in which it lies. Please identify this epithelial tissue. You would be able to tell me transitional epithelial cells. This is again another photograph. This again is from the University of Kansas site. Again, transitional epithelial tissue in the urinary bladder. Now, another form of tissue that we talked about was squamous epithelial, specifically stratified squamous epithelial. Now, stratified squamous epithelial is squamous cells that are stacked upon each other. Now, remember, these are thin cells, but you can see how they're stacked upon each other, a little thicker at the bottom, but get very thin as you reach the surface. Again, stratified squamous epithelial, we see it lining the entranceways into the GI tract. For example, this is the lips. The following slide is stratified squamous epithelial in the vagina. I also could put up a slide that shows you the rectum, which would show you very similar patterns. So any of the surfaces that enter the digestive tract have stratified squamous epithelial, or any opening into the human body, such as the vagina, has stratified squamous epithelium. Now, this is stratified squamous epithelium of the skin. You also can see in this structure, you also can see that there's also a level of callus formation also on this tissue as well. So this stratified squamous epithelium also has another border on the surface of it that lets you know that it is consistent with skin. Again, the cells are stacked upon each other. They get thinner as you get toward the surface of the skin. But when you see this formation, this callus formation above the surface, you know that this is stratified squamous, and you know actually that it's the skin that we're talking about, because it's the only area of the body where you see this type of pattern. Actually, these stratified squamous that we see at the mouth and the lips, in the vagina and the anus don't have this callus formation over the top of it. The keratin layers here are not as significant. But again, this is all still stratified squamous epithelium. Another picture, this is obviously, when we talk about the integumentary system in more detail, this is obviously thick skin versus thin skin. Very thick callus layer has formed over the stratified squamous epithelial tissue. This is the surface above the layer of the stratified squamous epithelium. Again, the magnification here is not as high as we had in the previous slides, but it's still, the pattern is very typical of what you would see in skin. An added feature here, and we'll talk about these structures more when we get into the integumentary system, is a layer of melanin. Melanin is a pigment that's located in the deeper tissue structures of the skin. It's in the stratum granulosum of the integumentary system, and we'll get into more detail about that later. But again, we're looking at it primarily for the stratified squamous epithelium. Again, you can see the callus formations over the surface of the skin. This is, again, all of these slides are from the University of Kansas Medical Center. And again, this is posted on the blackboard, the site that you need to go through and look at some of these selected slides as part of your lab assignment. But these are nerve cells. These are a typical picture of nerve cells in cross-sectional view in a histological slide. Again, you see the cell body, the axonal process, the dendrites that you can see located here as well. All a part of the nerve cells. Should be able to recognize this, identify them as either neurons or nerve cells. This slide illustrates skeletal muscles. Look at the longitudinal pattern that exists here. Also look at the longitudinal pattern that exists here. Also look at the pattern of striations, the peripheral lying nuclei. This pattern is very consistent with skeletal muscle. Now remember, there are ways to differentiate the striations of skeletal muscles from the striations of cardiac muscle, and I'll show you some upcoming slides of cardiac muscle, so you should be able to determine and differentiate the two. Skeletal muscle attaches to bone. It carries out locomotion in the body, carries out movement. Now this is smooth muscle. You notice the absence of striation in the smooth muscle. You also see the similar longitudinal pattern, but you don't see the bulkiness as you saw in the skeletal muscles. Remember, skeletal muscles are under certain types of stressors. They help in supporting the body weight. They also can develop, and so they hypertrophy more and they become thicker, whereas smooth muscle is not under those types of stressors, so the fibers are very thin. Still longitudinal, still peripherally lying nuclei, but you do not see the pattern of striations. Smooth muscles are located in the intestinal tract, the lungs, so in the respiratory center and the reproductive center as well. Primarily for contraction. Now this is cardiac muscle. Differentiate the cardiac muscle in comparison to the skeletal muscle. First, we see these structures where the blue arrow points, intercalated disc. These are the intracellular connections that we see between the cardiac muscle cells. Okay? Very, very prominent here. See, there are some other slides that will make this even more prominent. Also look at the branching of the muscle. Remember, the skeletal muscle was longitudinal. This has several branches that come off. Also, please note the location of the nuclei. A little more prominent, not as peripherally lying as you saw in the previous slide. You also see a little bit of fat cells here that are located in between. This slide is of cardiac muscle. Another slide indicating the intercalated disc. And then this is a different stain, a purple stain. You can see very prominent intercalated disc on this slide, still the branching pattern of the muscle, the fat cells as well. Again, the significant striations, but differentiated from skeletal muscles by these intercalated discs and the branching pattern. This slide is of the osteomes, or bone cells. Again, now it's labeled Haversian canal. Actually, that's the Haversian canal where you see the arrow. But basically, these are osteomes. And I would expect you to recognize this and identify this as bone tissue, osteome as well. If I ask you to identify this central structure, I would expect you to give me Haversian canal. You can see the concentric lamella that is very typical of bone tissue. This is an up-close picture, again, from the University of Kansas site. Again, if I asked you to identify this central structure, you would tell me this is the Haversian canal, a structure that runs through the Haversian canal, blood vessels. Concentric lamella that you can see, kind of looks like the inside of a tree. This is an individual osteome. Again, bone tissue. This center is the Haversian canal. This picture shows you a tendon made up of dense, regular connective tissue. If I asked you what is the tissue type that makes up a tendon, I would expect you to tell me dense, regular connective tissue. I would probably ask you that question just as I've just stated, because there's a similarity between this and smooth muscle. If you remember smooth muscle, though the stain was a little bit different, it also gave you this same wavy-like pattern. The muscle fibers are very thin, and so it's kind of hard to differentiate the two between dense, regular connective tissue and smooth muscle. I would have to be a little more specific in the description. Now, this is the tissue that lies inside organs such as the liver, the spleen, lymph nodes. This is reticular tissue. Reticular tissue. Specific stain, you can see the reticular fibers here, very typical of reticular tissue. More of a structural support than we see with some of the other types of tissues that we've been looking at today. Reticular tissue. And this final slide is of adipose tissue. The adipose tissue is actually these clear staining areas. Remember, adipose tissue does not pick up stain well. There are some special stains that you can use to help pick up adipose tissue. You can see one peripherally lying nuclei here, but it's very difficult to see the nuclei in these particular types of cells because they just don't pick up stain well. This is adipose tissue. Now this is an example of blood vessels. When we talk about the circulatory system in A&P2, we'll revisit this slide. But because of the thickness of this, we know muscle is inside this blood vessel, so this is an artery. Arteries have thick layers of muscle that make up the layers of the blood vessel, so this is an artery. This is a vein that lies next to it. Veins don't have the significant muscle layers like the arteries do, and so it's a much thinner lumen. But this is the lumen of a vein, the lumen of an artery, and again, this is the adipose tissue. Adipose tissue insulates, provides cushion for the body, also an energy source as well. So this is adipose tissue. This ends the histological slide presentation. Familiarize yourself with these slides. You should also familiarize yourself with the University of Kansas site, which I have posted for you on Blackboard. Thank you very much.